Assalamualaikum and hi everyone. I am Miss Siti Sarah Azman, and in this video I am going to introduce you on a process called protein synthesis. In the previous video you have learned about the process of DNA replication. Now I am going to briefly explain about other processes involving the DNA molecules. At the end of this lecture, you should be able to briefly describe how protein is being synthesized. Okay, so how you use the information in the DNA to produce protein, and you should be able to compare the differences between DNA and RNA molecules, and also you should be able to compare the three types of RNA molecules. Okay, and what are their functions in protein synthesis. Before we go further in this topic, let's just recap on what we have learned in the first chapter during the start of the semester. We've learned about enzymes, what are their functions, what they are made of. One example of enzymes, we have amylase. We use a lot of enzymes in the cells because enzyme catalyzes molecules, okay, and they are made up of proteins. But do you know how your body produces these proteins or those enzymes? And when your body, or specifically your cell, wants to produce these enzymes and these proteins, they need information to synthesize a molecule. So where do they get the information? For example, how do they get the information to produce amylase enzymes? And what about the other proteins? How they are being produced? So the answer lies in our DNA. Okay, and this is what we call as protein synthesis. We know that DNA is the blueprint of the cells, okay, because it contains the information. The information in the DNA is important because it is the information needed to produce these molecules. Okay, what are the molecules? Those are protein. Why does the information in the DNA only contains information to produce protein? What about other molecules? What about carbohydrates? What about lipids? Don't DNA contain information to produce all of these molecules? Okay, the answer is no. The information in the DNA is only used to synthesize protein. Why? We know that protein performs various functions in cells. Okay, for example, they act as structural support. For example, we have keratin protein, and some of the proteins act to transport molecules, okay, from in and out of the cell. And we have protein channels, we have protein carriers, and we have one group of protein that act as enzymes. This is the important one. Enzymes speeds up chemical reactions. What are the chemical reactions being speed up by enzymes? Enzymes can catalyze a reaction when you want to build larger molecules, when you want to combine monomers to build polymers. For example, you have carbohydrate monomers, you want to combine them to produce polysaccharides. Or enzymes can also catalyze breaking down large molecules into their smaller components. Okay, for example, when you eat your food, okay, so you eat carbohydrates, and enzymes will catalyze the reaction by breaking down carbohydrates into their monosaccharides. Okay, we know that almost all processes in the cell involve enzymes. When you want to build carbohydrates, when you want to break down carbohydrate molecules, when you want to build protein, or you want to break down protein, or when you want to produce lipid, or when you want to break down lipid into smaller molecules. Okay, so those processes involve enzymes. So that's why the information contained in the DNA codes for protein. Okay, because if you have protein enzymes, you can build other molecules. You can build carbohydrates, you can build lipids, you can build other proteins, you can break down those molecules. Okay, because if it codes for proteins, you can get other molecules as well. Before we go further into the topic of protein synthesis, okay, so these are some of the important terminologies that you have to know. The first one is chromosome. Okay, chromosome is a structure where you have your DNA molecule being packaged and being condensed so that you have this structure that we call as chromosome. If you untangle your chromosome molecule, you will actually find your DNA strand. And the other terminologies is DNA. Okay, DNA is this double-stranded nucleic acid that contains information to produce protein. Okay, so those informations are being formed in the sequence of nucleotides, okay, where you have this base pairing. And then we also have what we call as gene. Gene refers to functional segment of the DNA where it contains information to produce protein. Not all parts of the DNA contains information to produce protein. So the part where you have the genes, that is the part where it contains information for protein synthesis. For example, if this is gene, okay, this is the information to produce one protein or one polypeptide, and you might have this part which is also a gene that also contains information to produce maybe a different protein molecule. So gene refers to segments in the DNA. The DNA will be coiled and condensed so that it has this structure which we call it as chromosome. And then the last terminology, when people talk about gene expression, it refers to the process where you use the information in the DNA to produce protein molecules. We have the information in the form of gene, and then you want to express them into products. The product is protein. We know that gene expression is the process of how you want to use the information in genes to produce protein, but this is not a one-step process. So gene expression involves two stages. The first stage is what we call as transcription. So the process of transcription is where you take the information from the DNA, you copy them into another form. We call it as mRNA. The second stage is once you have the information in the form of mRNA, you need to convert this information into protein molecule, into amino acid sequence. Okay, so this stage we call it as translation. So the first process is transcription, the second one is translation. This picture shows the overview of protein synthesis that happens in eukaryotic cells, okay, which happens in our cell. In this picture, you can see that the first step in protein synthesis is transcription, and after that there is additional step, we call it as RNA processing, but this process is not being covered in our course. The final step in protein synthesis is translation process. So what is transcription? Transcription is the process of copying parts or segments of the DNA that is important for protein synthesis into a different form. Okay, that form is what we call as mRNA or messenger RNA. When protein is being synthesized, we did not copy the whole DNA just to produce one protein, okay, because our DNA is very long. Just to give you an idea of how long our DNA is, one of our chromosome contains around 260 million nucleotides. So in this one double-stranded DNA, there is 260 million nucleotides. And in this single DNA strand, there are about 2,000 genes, where you have around 2,000 informations that codes for protein. Okay, for example, we have gene number 1, gene number 2, gene number 3, up until 2,000 genes. Imagine your cell only want to produce one protein. Instead of copying the whole part of the DNA, what happened during transcription is information copied is only on the segment needed for protein synthesis. Okay, for example, if this is the part that contains information to produce protein A, this is the one that will be copied into mRNA during transcription. You can also imagine it as you have a Campbell Biology reference books. Okay, so that book is about 1,000 plus pages, and you only need information from one page in the book. So instead of photocopying the whole book, you only copy the page that is important to you. So that is how transcription works. When you want to produce one protein, the gene that codes for that particular protein is the one that is being copied into mRNA. And why do we need to copy this information into mRNA molecule? Okay, because DNA is located in your nucleus. Okay, so this is your nucleus. And then protein is being produced in the organelle called ribosome. Okay, so you have covered this during last semester. And ribosome is an organelle that is located in the cytoplasm. DNA cannot be transported out from the nucleus. So what happens is you copy the information into mRNA molecule, and this mRNA will be transferred out from the nucleus to the ribosome. So that is where you have RNA processing, but we are not going to cover that. Once the mRNA being transported to the ribosome, that's where you have translation process. mRNA is a nucleic acid. When you want to produce protein, protein is made up from amino acid sequence. So you will see that the language is not the same. Okay, mRNA is made up from nucleotides, protein is made up from amino acids. So what happened during translation is the mRNA will be translated into amino acid sequence, and then you will have your polypeptide chain, which will later become protein. This is another way to view protein synthesis. Okay, so what are the differences between DNA and RNA? Okay, so DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acids. So there are several differences between these two molecules. I think you have covered this before, okay, but this is just to refresh your memory. So some of the differences are the number of strands. DNA is double-stranded, and these two strands are in anti-parallel, while RNA molecules only contain one strand. Okay, so it is a single-stranded nucleic acid. There is no base pairs in RNA molecules. Another difference is in the form of nitrogenous bases involved. When you want to build DNA molecules, okay, so the monomers are nucleotides. Nucleotides contain phosphate group, pentose sugar, and also nitrogenous base. There are 4 nitrogenous bases used to build DNA molecules and also RNA molecules. In DNA, we have thymine, adenine, guanine, and cytosine nitrogenous bases, while in RNA we have uracil, adenine, guanine, and cytosine. So the difference between these two is thymine and uracil. We have A, T, C, G for DNA molecule, and then we have A, U, C, G in RNA molecules. Another difference is in their ribose sugar. Because of the name deoxyribonucleic acid, the ribose sugar in DNA is what we call as deoxyribose, while in RNA we have ribose sugar. So what is the difference between these two? Deoxyribose, they contain one less oxygen in the molecule. Another difference between DNA and RNA is their form. Okay, so you can only find one form of DNA. Okay, they are double-stranded, they are anti-parallel, and that is the only form of DNA you will find in the cell. However, you can have 3 forms of RNA molecules. Okay, so those 3 forms we call it as mRNA, tRNA, and also rRNA. We will look at the differences between these 3 RNAs later. And the other differences is their locations. DNA can only be found in cell's nucleus, cannot be transported out from the nucleus. However, RNA can be found in the nucleus or also in the cytoplasm. So this is the comparison in terms of structures. Okay, so you can look through. From the differences table before, we know that there are 3 types of RNA. Okay, so those 3 types of RNA are mRNA, tRNA, and also rRNA. The first type of RNA is what we call as mRNA, or messenger RNA. It is a linear single polynucleotide, and the function of mRNA is it carries the genetic code from the DNA, okay, because mRNA is the copy of that small part of the DNA during transcription process. The other types of RNA, we have rRNA, or we call it as ribosomal RNA. rRNA is the type of RNA that is used to build ribosome organelles. So this rRNA is being synthesized in the nucleolus. Ribosome is made up from rRNA and also protein molecules. So the function of rRNA, it is the same as the function of ribosome. It becomes the site where protein is being synthesized, or polypeptide is being synthesized. And the last type of RNA is what we call as tRNA, or transfer RNA. Transfer RNA, it's a single-stranded nucleic acid. It's just that they are being folded to become this cloverleaf shape. If you untangle them, it is still single-stranded. What is the functions of tRNA? In protein synthesis, specifically in the ribosomes, when the cell wants to build protein molecule, they have to combine amino acid together. So the function of tRNA is they are the one that helps to carry amino acids and bring it to the ribosomes. And if you see from the structure of the tRNA here, they have two sites. So one site attached to amino acid here, this is the part where they carry amino acids, and the other side here, okay, or we call it as anticodon. You will learn about this when you learn about translation process. So we know that there are three types of RNA, and these three types of RNA, they all work together in the process of protein synthesis. So what is the connection between these three types of RNA? So they have these cooperative functions in protein synthesis, okay? So once transcription process happen, you have the mRNA. The mRNA, since it is a copy from the DNA, it contains information to produce protein molecule. And when protein need to be synthesized, you need transfer RNA, because transfer RNA is the one that will carry amino acids. So the process of protein synthesis happens in the ribosome, in the ribosomal RNA. So this is the connection between these three RNA molecules in protein synthesis. So this is just an overview. So you know that protein synthesis happens in two stages. Okay, so the first stage is transcription. Information from DNA copied into mRNA. mRNA is being transported out from the nucleus into cytoplasm. This mRNA is being carried to the ribosome, or to the rRNA. And when protein synthesis happens, we have tRNA that carries the amino acid needed to build protein molecules. So I hope you understand about this. Further explanation about transcription and also translation process will be covered in the next two videos. And thank you. Bye.